Hi, let's talk about blood types. In this video, we'll talk about blood group systems, in particular the ABO and RH blood groups. We'll talk about their genetics and the ramifications of these blood groups. So blood group systems are based upon the surface membrane proteins of red blood cells. In this case, we refer to them as surface antigens, and antigen is a substance that generates an antibody. Antibodies are produced by plasma cells, which are derived from B cells, and they look like Ys. And the base of the Y is the more constant portion of this molecule, whereas the distal arms of the Y are known as the antibody binding locations. So we refer to these as paratopes, which are antigen binding sites. So these paratopes, or these ABSs, they bind to epitopes of antigens. So they can connect. Now, whenever you have an antigen plus an antibody, you're going to have an immune response. That immune response can be a variety of things. There is what's known as the classical pathway, which involves molecules known as complement, which destroy cells in the vicinity of the antibody-antigen complex. And then there's also something called agglutination. And with agglutination, you can have a single antibody connect to items through having similar epitopes. So in the case of anti-ABO blood group antibodies, you can have an antibody connect to erythrocytes. And when this happens, they agglutinate. They form this blob, and that's going to cause problems within the vasculature. Now, there are hundreds of recognized surface membrane proteins on erythrocytes, and these are organized into 36 different recognized systems. Of these 36 recognized systems, the two most popular that we hear of are the ABO group and the RH group. With the ABO, you can either be type A, type B, type AB, or type O. Now, these types are enumerated here. So for an individual who is type A, if we're looking under a light microscope, which we would not be able to see antibodies here or these surface membranes, but in this theoretical construct, here is our erythrocyte, and the erythrocytes are covered with these A antigens. And these antigens are actually glycoproteins, so they're associations of proteins and lipids. Now, all of the erythrocytes are covered with these A antigens. And within the blood plasma, we're going to see antibodies that have antibody binding sites, or paratopes, that will bind and destroy the B antigen. So people who have type A blood, their plasma contains anti-B antibodies. With type B blood, their erythrocytes have the B antigen on the surface, and they have anti-A antibodies within the plasma. People who are type AB have both A and B antigens on the surface, and they have no antibodies in the plasma. And that makes sense because if they did, their own antibodies would destroy their erythrocytes, which would be bad. People who are type O, O was originally meant to be 0, not the letter O. They have no, hence 0, A or B antigens on the surface of the red blood cells, yet they have both anti-A and anti-B antibodies within their plasma. And let's see where this comes into play. So this top row is everything that we've seen already. We know how the major players work. Type A has the A antigens and anti-B antibodies. Type B blood has B antigens and anti-A antibodies. Type AB has both A and B antigens with no antibodies against A or B. Type O has no antigens, but it has both anti-A and anti-B antibodies. So let's take a look at what can happen when various blood types donate to one another. So for starters, along this top row, that's going to be the recipient. And this bottom row will be donor blood. So for instance, someone who is type A, so they'll have A antigens on the surface and anti-B antibodies. If they were to receive type B blood, their antibodies would agglutinate it. Now, the anti-A and anti-B antibodies are a class of antibodies called IgMs. Ig is short for immunoglobulin. So all antibodies are immunoglobulins. So these are types of blood proteins. IgM is the largest type of all of the antibodies. It also happens to be the first on the scene, and it can work either through that classical pathway of involving complement and destroying everything around it, or through agglutination. And in the case of A and B antigens, IgM works through agglutination. And so oftentimes IgM antibodies are called agglutinins for that reason. So if you give someone who is type A type B blood, there's agglutination. And it's a deadly combination. Likewise, someone who is type B type A blood, again, for the same reasons, there's agglutination. If you give someone who is type AB any of the blood types, there is no agglutination. That is because someone who is type AB already has both A and B antigens, and they have no antibodies to agglutinate. That's why we would refer to type AB blood as the universal recipient. They can get blood from whomever. Now let's look at type O. Type O, as you recall, has no surface antigens, but it has both anti-A and anti-B antibodies in the plasma. And as a result, if type O receives blood from any type A, type B, or type AB blood, there's going to be agglutination, and that would be bad. So the only donor that type O will work with is another type O. So type O is good. Yet type O, because it has no surface antigens, can donate to type A, to type B, to type AB, and to type O. And as a result, we refer to type O blood as the universal donor. All right, let's switch gears and look at another blood group. This is the RH factor. Now, you probably have heard of blood types like A positive or A negative or O negative or AB positive. That positive or negative is in reference to the RH factor. And the RH factor has many different potential epitopes of different letters. The one in question in particular with the ramifications of health that we care about is the D factor. The allele coding for D is dominant. In order to be Rh negative, you would have to lack both D alleles. To be positive, you would only have to have one, or at least one. And these antigens, like ABO, are integral membrane proteins. People do produce antibodies to these antigens, and it's really just an issue of individuals who are Rh negative. Now that Rh, this group was originally studied in rhesus monkeys, and so in honor of that, this is the Rh group. So individuals that completely lack the D antigen are Rh negative. Worldwide, this is a small minority of people, and it does vary by geographic region. But worldwide, about 94% of individuals are Rh positive, and about 6% are Rh negative. So Rh negative is a very rare condition. Yet someone who is Rh negative is capable of generating anti-D antibodies. These anti-D antibodies are largely of a different class. They're predominantly immunoglobulin G. Immunoglobulin G is much smaller than IgM. IgG is probably the largest class of immunoglobulins, and it's the only antibody that's capable of crossing the placenta. That's going to have a couple of important ramifications. For starters, maternal IgG antibodies crossing the placenta are the basis behind passive immunity. Meaning, IgG antibodies in the mother crossing into the fetus's blood can confer specific immunity within the fetus for a short period of time. So this is adaptive immunity that is not based or predicated on the fetus being exposed to something. It's gained that immunity from its mother. But these antibodies, they don't last a terribly long time, so this passive immunity doesn't last a long time. Now, in the case of an Rh negative mother, if she were to conceive a child with an Rh positive man, so let's do Rh negative mother with a positive father. Depending on the actual genotype of the father, you can either get, a heterozygote or a homozygote here. And in the case of the homozygote, the baby, or there's, I'm sorry, in the case of the homozygote, the baby is Rh negative. In the case of the heterozygote, the baby is Rh positive. Now the issue here is that an Rh positive fetus can generate an immune response, so the mother can make anti-D antibodies against the antigens on the surface of that fetus's red blood cell. And what generally happens is, over the course of a pregnancy, some of that fetal blood leaks back across the placenta and can generate those anti-D antibodies. And that's very problematic for both that pregnancy and any subsequent pregnancy, because whenever the second pregnancy comes along, or I should say if ever a second pregnancy comes along, those anti-D antibodies can cross the placenta and they will attack the fetal red blood cells as if they are some foreign pathogen. This is known as hemolytic disease of the newborn. Hemolytic, so lysis of blood cells of the newborn. And there can be both agglutination and or hemolysis. So the erythrocytes can clump or they can be lysed. The good news is that there is a very effective prophylaxis for this, and it is an intramuscular injection called RhoGAM. RhoGAM is indicated for Rh negative women who are pregnant at about the 28th week of their pregnancy. It's an intramuscular injection, and it prevents the production of anti-D antibodies, therefore preventing any subsequent occurrence of hemolytic disease of the newborn, which is a wonderful thing for science. Now, a question for you to assess, if you've been paying attention along the way. So, for which blood type would it be the easiest to find a transfusion donor? Would that be type A, type AB, type B, or type O? Well, let's think about this. Type A can receive blood from people that are type A or people that are type O. Type AB can receive blood from A types, B types, AB types, or O types. Type B can receive blood from B or O, and type O can receive blood only from O. So the easiest blood type to find a transfusion donor would be the universal recipient here, type AB. Thank you very much for your time.